Welcome to OmicsBox, Bioinformatics Made Easy.
Okay, so in this first part of the webinar, we will focus on the definition and curation of long-read transcriptomes with OmicsBox. First of all, it is important to ask ourselves why long-read sequencing is important in transcriptomics. And the fact that is that long-read sequencing was declared method of the year 2022 by Nature Methods. And the answer to our question is that whereas with short-read RNA-Seq, you need an assembly step to define a transcriptome, with long-read RNA-Seq, no matter if it is PacBio or ONT, it is possible to capture full-length transcripts inside one single read. So, I'm going to talk about a bit very quickly about our dataset. The species that we are going to use is the grizzly bear, and our data is composed of 6 PacBio full-length non-concatemer reads that were obtained using PacBio SQL systems, and 12 pair-end short reads files that were obtained from Illumina HiSeq. The experimental design is half of the dataset is from active adipose tissue, and the other half was obtained from hibernation adipose tissue from the grizzly bear. This dataset was obtained from this paper. It is really interesting. I recommend you to read it. I like it. Okay. I want to summarize a bit how PacBio sequencing for transcriptomics work, in case anyone needs to remember it. And first of all, the RNA is reverse transcribed into cDNA, and then it is circularized thanks to these primers, and then the smart bell structure is formed. Then the polymerase reads this smart bell and generates the so-called subreads. That is a set of full-length or passes of this smart bell. Then using the IsoQuant workflow that it is already available in OmicsBox, you can go from these subreads to the CCS. That means circular consensus sequence. Then from this circular consensus sequence, you can go to the FLNC reads, the full-length non-concatemer reads, that are the ones that we are going to use. You can obtain them also using the IsoQuant workflow. Okay, so the pipeline that we are going to follow in OmicsBox is, first of all, a long-read quality assessment using LongQC. Then we will define our transcriptome with FLAIR. And finally, we will create and filter this long-read transcriptome using scanty3 in order to have a curated and filtered transcriptome model that you can use in downstream analysis. So now we move to OmicsBox. And as we have said, the first thing that we are going to use is LongQC to assess our long reads. So we click on General Tools, FastQ Tools, and Long Reads Quality Assessment with LongQC. It is true that you can't you can use as input not only FastQ files but also FASTA files, or even the PacBio BAM files. So you add all your files of your dataset, all your samples. Then you select the sequencing technology that has been used. In our case was PacBio SQL, as we have mentioned. Nevertheless, you have other PacBio technologies and different ONT sequencing kits. Then, well, yeah, we have to enable the transcript mode because we know that we are sequencing or analyzing transcriptome models. And we don't select the short mode because we don't have short reads and the save trim reads option because we are not going to trim reads and trim and save them. So we just click on run. Well, not in my case because I have already run this analysis and I have it here. Yep. This is what you will see once LongQC has finished. As you can see here in this tags column, all of our long reads samples in our dataset are correct. That means that they don't have any kind of error or warning, and they are safe to be used in subsequent analysis. And you have also here some metrics that you can see them here better. Yeah, you can see that we have different metrics, and as you can see, all of them have similar numbers, like they have homogeneous metrics. That's good. That means that there are no samples that have outliers or strange things. So it seems that our dataset is good. Nevertheless, maybe we can look also. We can click on charts, open the sequences composition, and yeah, we select all the samples, and we select these two plots. Yeah. So these two plots are opened here in OmicsBox. The GC content plot shows the distribution of the GC content in the reads of the different samples. And it is obvious that if we have sequenced the same organism, this distribution might be similar, and it is. So it is good news. And in the masked basis, sorry, again, the low complexity fraction must follow a similar distribution in all the samples, and that's also what is happening here. So yeah, it seems that all of our samples are good. We can also open the reach length distribution chart. We just select all the samples, and again, all the samples have to follow the same or more or less the same reach length distribution as it is happening here. So yeah, we can say that we can go on with our analysis. So once that we have assessed our long reads, we can generate our transcriptome model. To do so, we click on transcriptomics, long reads analysis, transcript identification, and we're going to use FLAIR. You can also use IsoQuant, IsoQuant, or this reference-free pipeline. But in the longer gasp research that has been published not long ago in Nature Methods, and in which Biobam has contributed to, in that research, they found out that FLAIR is one of the best algorithms to do the identification of isoforms. So we are going to use FLAIR in this case. Okay, so you have to add obviously all your samples of your dataset because we want to generate a transcriptome with transcripts of all the samples. And then you have to add the reference genome and the genome annotation in my case. Yeah, it is true that the grizzly bear, it might not be the best studied organism. It is not a very well studied organism, but it's true that we have the reference genome and the genome annotation available in different repositories. But imagine that you are working with an organism that you only have the reference genome. So you would click in, well, yeah, you wouldn't select this option, And as you can read here, you will need the short read information. So you will select this option of use short reads. And yeah, you can add the BAM files or the tab files that can be also generated inside OmicsBox. To do so, you just have to add the short reads FASTQ files you click in RNA-Seq and you add the FASTQ files to start. You add here the FASTQ files and you will obtain the BAM files and or the tab files that you can use there. Yeah. Also, imagine that you are working with a very unknown organism that you have sequenced with long reads its transcriptome, but you don't have neither the reference genome or the genome annotation. So in that case, you wouldn't be able to use FLAIR, but you can go to transcriptomics, long read analysis, transcript identification, and use this reference-free isoform reconstruction pipeline that it's called ISOM pipeline. You will only need the long read files, but in this case, you have to use it in FASTQ format because the quality of each nucleotide is needed. So you have to add here FASTQ files. Okay, nevertheless, we will continue with our analysis. We were using FLAIR, and we were using these long reads in FASTA format, the reference genome, and the genome annotation. We'll click on next, and you will see this another wizard page in which you can add your own alignment files, the aligned reads in BAM format from your long reads. Long reads can also be aligned to a reference genome using Minimap2 inside OmicsBox. But this option might be an advanced option because maybe it's just in case you want to change some default parameter in Minimap2, or maybe you have the BAM file from another aligner and you want to add them here. But in this case, we won't select this option and we let FLAIR to use Minimap2 on default parameters. Also, you can click here if you want to quantify reads and add a manifest file that is something similar to an experimental design file. But by now we don't want to quantify reads. So we just click on next. Here you have the different steps of FLAIR. I will run FLAIR on default parameters, but I will also make two changes. These are the default parameters, and I want to change these two. I want to treat redundancy. Later on, we will talk about redundancy in transcriptomics. By now, I will just select that I want to treat redundancy by maximizing the transcript start site and transcript termination site. Also, I want to filter isoforms inside FLAIR. I want to select a more stringent option in which I will filter isoforms based on support, based on subset isoforms, and also I want to remove single exon isoforms. It is true that, like in the transcriptome, there are single exon isoforms in transcriptomes, but it is true that sometimes when we sequenced long reads with long reads a transcriptome, some single exon isoforms might appear as a consequence of a truncated polyexon isoform. So by now I want to remove them. So I click on next, and I just have to select the directory or the path where I want to store the transcriptome annotation in a GTF format, transcriptome sequences in FASTA format, and this file, the isoform read map. this map.txt file has the information about which long reads were used to generate which transcripts. Also, we have the counts file that in this case is not available because we said that we didn't want to quantify transcripts by now. So yeah, you would click on run again. I will click on cancel and I will open my analysis. It is here. This is what you will see in OmicsBox once FLAIR has finished. As you can see here, you also have the GTF file with the transcriptome model in this format, then the sequences in FASTA format, and even the map.txt file. Because this was the directory where I stored this information. Okay, so in this summary report, you will see first of all the input data, the file names that you used, and some summary metrics of your transcriptome. The number of isoforms, the average length of your transcripts, and the minimum and maximum length. This is a very short transcript and this is a very long transcript. So as you can see, maybe we will have to filter out some isoforms later. Here also you have information about the correction step and the collapsing step. Here you can see the long reads that were rejected in this step and the ones that were valid for subsequent steps. These subsequent steps, in this case, is just one, the collapsing step. Here you can also see the isoforms that appear after the collapsing step and how many long reads have been collapsed to make transcripts. This is the absolute number and this is the relative one. And yeah, also you can see here the parameters that we have used, maybe just to keep reproducibility and just in case you want to repeat the analysis with another parameters. And yeah, this is the isoforms length chart distribution. As you can see here, the x-axis is a bit strange, but it's because of the long transcript that we saw. You can deselect this option and yeah, you can see it better now. Okay, so we first of all assess our long reads. Then we made our transcriptome model, and now we have to curate it and filter artifacts. So yeah, to do so, we just click in transcriptomics, long reads analysis, and curation of long reads transcriptomes with scanty3. In this first page of the wizard, you can add the minimum input that you need. This is the transcriptome. In this case, you can select or transcriptome in GTF format, the annotation file, the sequences in FASTA or FASTQ format, or this option that it is not really recommended. Because in this option you will add the raw long reads and then a collapsing step will be done before running scanty3 in order to make some kind of transcriptome, but obviously it is better to use or FLAIR or IsoQuant, IsoQuant or the ISO pipeline before using this option. And between these options, I would say that I will recommend to add the transcriptome in GTF format, because if you add the sequences, that the results must be the same. The thing is that an alignment step is going to be done before the scanty3. So you will lose more time will be consumed. So if you have the GTF file and the FASTA file, that it's something that happens in IsoQuant or in FLAIR, you just add the GTF file and that's fine. Also, you have to add the reference genome and the genome annotation. And you just click on next. In this second wizard, you will see that you can add short read information, in this case in FASTQ format. And this is something that is highly recommended. That's because this short read information will be used in the filtering step to validate splice junctions. And in addition, it is not necessary that these FASTQ files, these short read FASTQ files, come from the same experiment of your long read analysis. Maybe you have sequenced, well, imagine in this case, we have just sequenced the beer transcriptome using long reads. But we didn't use short reads, and we didn't, if in case that we didn't have these short reads, we can just go to a repository and download short reads of beer, and we can use it. We can use them here. It wouldn't be any issue. In this case, we have short reads from the same experiment, so we add them and we click on next. In this page, you can see also more additional input. These files are common for organisms that are very well studied, like human or mouse. It is not our case, so we just leave this wizard on blank and we click on next. And here you can see the parameters for the different steps in scanty3. Here is the first step, the quality control. In this step, scanty3 just will differentiate or classifies your transcript inside the different categories that it has. We will talk about them later. And once that they are classified, it will start the second step, the filtering step. We have three different filtering, or scanty3 has three different filtering methods. The first of all is the machine learning, that it uses a random forest to classify or to differentiate long reads, transcripts that are actually artifacts from real transcripts. And then we have these other two filters that they are quite similar, because the legacy filter is the rules that were applied in older versions of Scanty. So this is the oldest Scanty3 filter, and it is what the one that we are going to use. But now you can also add custom rules by yourself. You can add a JSON file with some custom rules. Nevertheless, as we have said, we will use the legacy filter, and we click on next. This is the last step of Scanty. It is the rescue step. It runs after the filtering step, and it is used to rescue isoforms that might be filtered out, but they seem to appear in your reference transcriptome, so you want to recover them. Nevertheless, this step do so as long as the redundancy is not raised. Now we are going to speak about redundancy in some minutes. So yeah, we just click on, we want to rescue transcripts, we click on next, and we just select where we want to save the information. We have some additional information files, the filtered and the created transcriptome annotation file in GTF format. Also, we have the sequences of the transcripts in FASTA format and the translated transcriptome. So you select a directory to save everything, and you just click on run. Again, I will click on cancel, and I will open the results. As you can see here, I have all the information that I have just mentioned, and here you will see this once scanty3 finishes in OmicsBox. In this summary report, you have a lot of information that is really useful. First of all, you have a gene level table, transcript level table, and junction level table. Also information about the filtering and rescue step. As you can see here, a lot of novel genes have been filtered out. In this column, you will see the number of novel genes that that the transcriptome had as it came out of FLAIR, and this is after the filtering step and rescue step. Obviously, we have just one novel gene, and in the at transcript level, we can see that a lot of these categories have been filtered out. These categories are likely to be artifacts, so it is good that a lot of them have been filtered out. Almost every isoform but one. And these other four are more common categories. The category that might have most of the transcripts, and it is recommended that something like this happens, is the full splice match. These isoforms are the ones that match a reference transcript at all junctions. Then the incomplete splice matches are something similar, but matches consecutive, but not all splice junctions. And then you have novel isoforms that this can, with this you can measure the degree of novelty of your transcript model. Then also you have junction level information and a filtering table and a rescue step information table. This table will be different if you use the rules filter or the legacy filter, or if you use the random forest filter. In this case, we can see the rules that were applied in the older versions of Scanty3. In order to keep full splice matches, they must have less than a 60% of genomic adenine downstream the transcript termination site, the TTS. This is because with more than a 60% of genomic adenine, you can say that this isoform might have happened because of interpriming. That is an issue that might arise when you sequence a transcriptome. That is because inside the transcript there are like a lot of adenine, so the poly-DT that is used to bind the polymerase, it might be it might be bound there. So it might be like some kind of a transcript that is actually an artifact. And then for the rest of the categories, you have to, they have to match all these rules in order to keep them. They have to, the same as before. They don't have to be an intrapriming event. They have all the space sites must be all canonical. Also, the splice junctions must have at least 3 short reads in them as with coverage, and also they don't have to be an RT switching artifact. This RT switching is something that also happens at the sequencing step, that maybe because of the secondary structure of the RNA, the polymerase might jump from one part of the transcript to another part of the transcript, or maybe it just jumped to another transcript. So some fusion transcripts or some truncated transcripts appear. So you also have to be aware of this and you can filter out isoforms that are actually truncated or fusion artifacts with this option. Then also you have, well, rescue information and the parameters, just in case you want to keep the reproducibility, or you want to change some parameters. Also, you can see this table that has a lot of information. Maybe in case that you want to remove something, you can just select something and remove it. This last column is the TSS ratio. It appears just if you used short reads, and it is something really useful because this ratio is the TSS ratio is the ratio between the coverage inside the transcript, the start site, and outside the TSS. So the greater the TSS ratio, the more probable is that the transcript is true, is real. So yeah, if for example you have something very low, maybe 1 for example, you can say that the coverage that you have inside and outside the transcript is very similar, so you cannot really be sure that that transcript has been registered by the short reads experiment. So you cannot rely on that transcript. Also, you have different charts. We will see some of them that might be useful. For example, you can see the distances of this of FSM or ISM, like full splice matches and incomplete splice matches, to the distance of them to the transcript termination site or the transcript start site. We will just select the FSM. We click on run. Well, the range is a bit strange, so we won't select this option. And as you can see here, they are very close to 0. Those transcripts start near or even inside an annotated TSS, and they finish in an also annotated TTS or transcript termination site. So you can be sure that full splice matches are reliable transcripts. You can also see the different reasons of transcript removal. The majority of transcripts have been removed because of the intrapriming problem, but also because of the RT switching problem, because not all the splice junctions were canonical, or even also because there was not enough minimum coverage of short reads. Finally, I want to talk about redundancy a bit. Redundancy is the problem of having actually the same transcript, but having it duplicated because of 3' and 5' variability. We want to have unique transcripts. Obviously, we don't want to have redundant transcripts. So in scanty3, you can do this kind of analysis. And as you can see here, the great majority of ISM and FSM isoforms are unique. So that's good. With all this information and what we have spoken about the different charts, the summary report, and so on, we can say that this transcriptome is really good and that it is curated and we can use it in downstream analysis. Now to finish, I want to share with you some conclusions. First of all, that the assessment of long reads is really important to initiate our analysis effectively. It is important to look, for example, at the length distribution or that the reads composition is homogeneous. Also, we have seen that OmicsBox makes it really easy to define your transcriptome because of the wizard. It helps you a lot to tune your parameters, also because of the summary report and the charts. And finally, also I want to remark the importance of assessing and curating your transcriptome using Scanty3 with all the features that we have spoken about. And yeah, it is important to create your transcriptome before moving on with downstream analysis. So thank you all for being here. It has been a pleasure. And now I will let Fabian continue with downstream analysis. And I want just to remind you the existence of the blogs in the Biobam webpage. I will paste the link to these ones in the chat now. So just in case you want to read about Minimap and the longer gasp research. And yeah, that's it. Thank you.
Thank you for watching. Learn more about OmicsBox with our other videos.